and I will start with the lecture 2 of signaling. Name some technique to analyze the signal transduction. The first one is enzyme immunoassay, which is known as ELISA, radio immunoassay, and high performance liquid chromatography, mass spectrometry, and the fluorescent or luminescent assay. So these are the techniques we use them to analyze the signal transduction. Now I go to the second questions is how do you explain bias signaling? We heard about something is called the bias signaling and what that's mean. The bias signaling is the ligand-dependent selectivity for certain signal transduction pathway relative to the reference ligand, often endogenous hormone or peptide at the same receptors. It seems to me it's when you just read this, you can't understand anything. Bias signaling is the ligand-dependent selectivity for certain signal transduction pathway relative to a reference ligand, for I can understand at the same receptor. Okay, I will explain that. If we just have two pathways, pathway A and pathway B. The pathway A, when activation of this pathway A, that is lead to some good things like signalings and other things, okay, good things. And pathway, when activation of it, that leads to severe side effects. Naturally, physiologically, normally, when there is a hormone that activate both pathways equally, that is lead to pathway that is lead to the activation, the good things, and in the same time, because it activation both of them equally, so that is lead in the same time the side effects, okay. But bias signaling, it means like this substance stimulate the pathway A because it leads to good thing and doesn't be handle the pathway B as the pathway A, okay. So that is lead to avoid the side effects, and that's exactly what is a bias signaling. Bias signaling occurs when a ligand molecule that binds to the receptor preferentially activates some pathways over others, like work in on pathway and leave the others after binding to the same receptors. And this is in contrast to a reference ligand, because the reference ligands activate both pathways in the same thing and equally. The reference ligand, such as a natural hormone or peptide, they normally binds to the receptor, which may activate all the pathways equally. And maybe this pathway that is lead to the good things and the other pathways lead to the bad things, like this. So the bias signaling is the ligand dependent selectivity. That means it's selective for certain signal the transduction pathway relative to the reference ligand. I mean the relative ligand reference ligands, it deals with the all pathways equally because it's natural, but the bias signaling, the something that we given the ligand is like be handled with the pathway not equally, the selectivity is not equally. Other terms we can say agonist trafficking, biased agonism, functional selectivity, ligand bias, or differential engagement, that we can call it like this. It's not always called the bias signaling. We can say agonist trafficking, we can say bias agonism, we can say functional selectivity, we can say ligand bias, we can say differential engagement. So we can say different names for this. Which physiological processes are controlled by beta adrenergic signaling? So we have different beta subunits like beta 1, beta 2, beta 3. Beta 1, it controls the heart rate, contractility, stroke, volume, excitation, conduction. Beta 2, bronchodilation, vasodilatation, glycogen lysis, that's meaning increasing the glucose in the blood, and it controls also the muscle strength. Beta 3, lipolysis, increase the lipid, thermogenic activity in the brown adipose tissue. And let's talk this and then we explain that is lead to the third questions, which is explain the process of thermogenesis in the brown fat tissue. Thermogenesis in the brown fat tissue is also known as brown adipose tissue, BAT, is a process by which the body generates heat, especially in response to the cold exposure. Unlike white fat, which stores energy. So, white fat is for energy, brown fat for heat, okay, for heat production. So, the brown fat is specialized for energy expenditure and heat production, and process is particularly important for thermoregulation in mammals and is driven by unique protein called uncoupling protein 1 found in the mitochondria of the brown fat cells. So behind all these processes is something is called uncoupling protein 1, which is present in the mitochondria that is present inside the brown fat cells. So we can see from this speech that there is two types of the fat cells, white fat cells and the brown fat cells. The white fat cells that is important for storage of energy, while the brown fat cells is important for heat productions. And all the process is driven by uncoupling protein 1, which is found in the mitochondria, which is that is present in a brown fat tissue. Okay, so the key steps in thermogenesis in a brown fat tissues. What has happened? The first one is called exposure and sympathetic activation. When the body is exposed to the cold, which is the cause, when the body is exposed to the cold, the sympathetic nervous system is activated, and that is due to the releasing of a neurotransmitter norepinephrine or noradrenaline. Norepinephrine then it binds to the beta 3 adrenergic receptors that is present on the surface of the brown fat cells, and that is initiating the thermogenic process again. So the cause is a cold exposure. The cold exposure activating the sympathetic nervous system. The sympathetic nervous system increasing the release of the norepinephrine. Norepinephrine binding to the beta 3 adrenergic receptor that is present on the surface of the brown fat cells. Then what has happened? Activation of the CAMP, binding of the norepinephrine to the beta 3 adrenergic receptors that is present on the surface of the brown fat tissue or cells triggers the G protein coupled receptors signaling cascade, and that is lead to the activation of the adenyl cyclase, and the adenyl cyclase that is lead to an increasing of the CAMP, which is known as a second messenger, and elevated CAMP that is lead to the activation of a protein kinase A, and kinase add phosphate, kinase, the word kinase, it add a phosphatic group which is phosphorylates downstream triggers, and that is leading including transcription factors and the enzymes that regulate the thermogenesis. Then the protein kinase A promotes lipolysis. That's mean the breakdown of its stored triglycerides fat that is present in the brown fat cells, and that is lead to releasing of a free fatty acid. These free fatty acids serve as a both fuel source and activators of uncoupled protein 1, which that's what we want. Uncoupled protein 1 located in the inner mitochondria membrane of the brown adipocytes, which is the key player in the thermogenesis, which is our player here. In normal mitochondria, the electron transport chain generates a protonic gradient across the inner membrane, which is used to produce ATP via ATP synthase. So the uncoupled protein 1 uncouples oxidative phosphorylation by allowing protons to flow back across the inner mitochondria membrane without producing ATP, and thereby that is leading to the dissipating the energy as heat. So the process of a protein leakage through the uncoupled protein generates heat, like the process of entering these or leakage of this proton through the uncoupled protein, that will be generates heat, which warms the body in this heat production is called non-shivering thermogenesis. Is critical for maintaining body temperature in cold environment. Wow. So I can mention this process and like steps. So the first one is called exposure and sympathetic activation, activation of ZAMP, lipolysis and the fuel mobilization, activation uncoupling protein 1, and then heat production. We make we make like a summary of thermogenesis. Brown fat uses stored energy to produce heat through the activation of uncoupled protein 1. The process is initiated by the sympathetic stimulation and that involved the release of non-epinephrine, the activation of the CAMP protein kinase A pathways, and that is lead to the mobilization of fatty acid and the subsequent activations of uncoupled protein 1 that is present on the inner membrane of the mitochondria, leading to the dissipation of energy as a heat instead of ATP production. It's a production of a heat instead of ATP energy that I mean here. So the ability to generate heat makes brown fat an important tissue for regulating the body temperature and has potential implication in the metabolic health and the treatment of obesity. So we end this lecture too. Thank you so much.